All right, we're excited. We're ready to move into our explore phase. And students, you are in your groups of 4. We're going to start off with the lab today, where we're going to be doing a teacher demonstration first. Our explore phase is going to have 3 different parts, and part 1 is going to be our teacher demonstration. So let's take a moment here and look at what we have. This is our lab setup. We have some resources, so chemicals here. We have resources that you have at your desk. So I know that you have your color pencils at your desk because you're going to be using them. Let me move that to the side. And you have your lab sheet also at your desk. And we're going to be answering a few little questions on our lab sheet as we prepare for our lab. All right, so let's looking at your lab sheet. You have definitely put your name and date on here, and we're gonna start with some pre-questions. Can anybody tell me what an indicator is? What is an indicator? Because we're going to be looking at BTB here, bromothymol blue, and it is an indicator. And we need to know exactly what an indicator is. Okay, we were introduced to indicators when we were doing our macromolecule lesson, and we were introduced to pH during our focus on enzymes. And BTB here, this is a pH indicator. So someone think about this. If this is a pH indicator, what exactly is an indicator? Okay, excellent. An indicator is any substance that gives a visual sign, usually by a color change, for the presence or absence of some concentration of a specific chemical, such as an acid or a base in a solution. When we were studying our macromolecules, we saw Benedict's solution, and we knew that it was an indicator because it tested for simple sugars. Such as glucose, and it's a clear blue solution. In the presence of a simple sugar, that blue solution changes color to green, yellow, and even brick red depending upon the amount of sugar. We use Benedict's solution in our cell component and macromolecules labs. Can somebody tell me what pH is? What is pH? We learned that when we were studying our enzyme labs. Okay, yes, awesome. pH measures hydrogen ion concentration. It's the measurement of the acidity or alkalinity of a solution. The pH scale ranges from 0 to 14. pH less than 7 is an acid. pH greater than 7 is a base, and 7 is neutral. And I know that if you didn't know, I would have refreshed your memory. However, you're right on it. So this BTB here, which is also already poured into here, we see it's a bluish color. It is a pH indicator. It will identify a substance if it's an acid. So this is called an acid indicator. This is blue, and if there's an acid present, it's going to change from blue to yellow. And it will get stronger and stronger in its color the more acidic the substance is. So we're looking for carbon dioxide to change this from the color blue to yellow, or some range of yellow. That will let us know that there's an acid presence. So we're looking for carbon dioxide, which is carbonic acid. Do we know of anything that has carbonic acid in it or lots of carbon dioxide in it? Are you aware that you drink carbon dioxide on a regular basis? Anybody can think of something that you might drink that has carbon dioxide in it? Soda and carbonated water. So I have some little things here to show you. This is a good old Pepsi. My dad loves Pepsi, so all I had to do was steal one of his Pepsis. He likes Pepsi with no sugar. And then this is my mother's drink. She loves this. This is a carbonated water, and we can tell here because it says flavored mineral water with natural CO2 added. So it has carbon dioxide in there. And let's see if it says anything about our sodas. Carbonated water. Okay, so carbonated water. So we know it has carbon in there. It has carbonic acid in there. Both of these do. And we're going to see if this pH indicator will really work. What do you think would happen if I poured a little bit of this Pepsi? Well, this is kind of dark, so we'll take a lighter color. Let's see if we had some of this carbonated water. What do you think would happen? Let's take a moment with your elbow partners and just tell your neighbor what do you think is going to happen. If we said this is the BTB was an acid indicator, it's blue, it's supposed to change colors in the site of an acid. Once I add this in here, let's see what happens. Let's look. Oh, what is happening? It's changing what? Colors. What color is it changing? Oh, the more I put in there, the lighter the color is becoming. It's almost a full can. Orangey yellow here. Wow, it changed colors. Since it changed from that dark blue to this orangey color, what does that tell me about this substance here? What does that tell me about this carbonated water? Where on the pH scale do you think that this carbonated water would be? Do you think that it is high, meaning above 7? If it's above 7, that means it's a base. Do you think it's below 7 as an acid, or do you think it is neutral? Do you think it's neutral right at 7? What do we think? You're correct. It changed because it changed in the face of an acid. And if this is an acid, which made it change from that blue to a yellow color, this BTB indicator means that this is in fact an acid. So that means its pH is less than 7. Less than 7. Good. Good, good, good. So now we're going to move this to the side just a little bit. And now we have something that's neutral. What do we know that's neutral? This is water. Came right out of this Nestle Pure Life water. And we said water was neutral. So it is 7 on the pH scale. So I wonder, will it cause this to change colors? Let's see. So on your lab sheet, we've identified our pre-question. What is the indicator? Let's think about, we've answered, why is this an indicator? Because it tests for what? It tests for an acid, and it will change colors in the presence of an acid. For number 3, what happened when we mixed the BTB and the soda or the carbonated water? It changed colors. And why did this occur? It occurred because it was in the presence of an acid, okay? So now let's see what happens when it's mixed with water. So we're gonna pour a little bit of this BCB in here. Maybe didn't need that much, but we're gonna try it anyway. I'm gonna put that, this is a funnel, put that in there. We're gonna take this glass and we're gonna pour some of this in there. What color is that? Did it change to a yellow? What did it change to? It really didn't. It stayed kind of the same color, didn't it? So let's talk about what happened. Write down for your response what happened. And I think we'll agree it stayed the same color. And why? Why didn't it change to another color? Somebody tell me, why do they think that this stayed the same color? This is water. We agree that water is neutral. It's a 7 on the pH scale. Why didn't this change colors? Thank you. Yes, so we know, we identified that BCB, this substance, is a acid indicator, meaning that it tests for acids. It will only change colors from the dark color to this light color in the presence of an acid. And is water an acid? Is it higher or lower? Is it a base? What is water? What is it? So we know that it is less than 7. So water, an acid is less than 7. However, this water is 7, so it's not an acid. So it's not going to change colors. Now let's see if there's something that I can put in here to make it change colors. So we already know that if we were to put some of this good soda, carbonated water, or even if we were to pour a soda in there, it already has carbonated water in it, so it will change colors. So we're gonna see if my own body can make it change colors. Am I magical like that? Am I a super scientist? So I have a straw. We took it out of the container so it's sanitized, and I'm going to take my funnel out, put it back over here. Now, I am not drinking this like you would in a regular drink or a milkshake, but I am going to breathe into it. And what's a good way to breathe into water? We do it all the time when you were a kid and you had milk. You would blow bubbles into your milk to see those giant bubbles come up. So now I'm going to blow bubbles into the water, and we're going to see if anything happens. Ooh. Guess I shouldn't blow their clothes on. Ooh, let's pour some of this out. It's a lot. Well, we do see some color change a little bit. All right, now let's try. Getting some color change, some. All right, so it is changing colors. Let's see if we can get it to change a little more. All right, I think we're getting to some yellow there. So why did it change colors from the dark blue to the yellow? It went from green, from blue to green to yellow. And I wonder why that occurred. Why do you think that occurred? So here we noticed that there was carbonated water. Carbon was put into here. And here I'm breathing out into the water. What am I breathing out into the water? I am breathing out carbon dioxide. And carbon dioxide is something that I am expelling. So on your lab sheet, you drew with your color pencils the before, what happened before, the dark color, the during while I was blowing, you drew your during, and then of course you drew what occurred. We have a yellow color now. So in your groups, have a discussion, and let's discuss the 3 questions on your sheets. When air was blown into the water mixed with BTB, what happens? Describe the color change with respect to the amount of blowing. At first, when I was blowing, what did you see happen? The more that we blew, what happened? And then, what chemical compound do you think caused that change? After your discussion, I want you to make a prediction with your partner. What do you think will happen if an aquatic plant is added to the test tube and left over time?